Welcome to Parasitic Diseases. Today we're going to discuss the Leishmania. We're going to begin with an introduction because this is a large group of organisms. They're found throughout the world, both in the New World and in the Old World. And it's good to get some biology under our belt before we start to look at the clinical diseases that they cause. So what you see here on the introductory page are two extreme examples of the diseases that this organism and the group of organisms known as Leishmania can cause. They have been with us since humans have lived on this earth. And they will probably be with us during our late stages of evolution when we are busy trying to extinct ourselves. But for the time being, what we're trying to do is identify the parasite, institute a therapy that is effective, and if at all possible, try to prevent reinfection. So without further ado, let's begin. So as I mentioned, the Leishmania are a complex group of organisms. All have the same genus name, but as you'll notice, there is a large number of species names associated with this organism. We have three main groups to discuss: the cutaneous Leishmaniasis group, the mucocutaneous Leishmaniasis organism, because there's only really one that causes that, and finally the visceral Leishmaniasis group. There are two that have been implicated in visceral leishmaniasis. As their names imply in terms of clinical manifestations, some are cutaneous, some take advantage of the mucocutaneous junctions in our body to initiate infections, and the last group, which is the most lethal of all of these, is the visceral leishmaniasis group, which infects basically the cells that are designed to protect us against infection to begin with. So let's look at each one separately to begin with, and notice that some of the genus and species of Leishmania are in red, some are in black, and a few are in black and red. The organisms designated in black type are found in certain parts of Europe, the Mediterranean basin, North Africa, the Middle East, South Asia, and Asia. Whereas those designated in red type are found throughout Central and South America. Many of them are zoonotic, that is to say, they come from animals that live adjacent to, or perhaps not so adjacent to, where people live. And they have all emerged over the last 20 years or so into names of species which correlate to the countries in which they were localized to begin with. So the next thing that we can look at is the geographic distribution of these in more detail. So of the major types of Leishmaniasis that we are going to concentrate our clinical and biological efforts on, Leishmania donovani rules as the number one cause of death due to this group of organisms. It's the visceral Leishmaniasis organism. There's another group that's insidious in the sense that they create disfiguring diseases, all related to Leishmania brasiliensis, and as the name implies, of course, it's mainly found throughout South America and certain parts of Central America. And then finally, Leishmania aethiopica and Leishmania major, which were thought to only be cutaneous in their manifestations. As it turns out, it depends on who catches it as to how these organisms present themselves. And this will become clear later on in the discussion. But first, a word about where these organisms fit into the grander scheme of things with regards to protozoans, because we're emphasizing protozoans in the first portion of our parasitic disease online course. So the first organism we discussed last time was Giardia lamblia. It had a very simple biology, living in the intestines, living not just in the intestinal tract, but just on this outside of columnar cells, reproduced by binary fission, had a cyst stage and a trophozoite stage. Very simple biology compared to the ones that we are about to discuss. This large group, called the KanetoPlastidae, consists of two groups of organisms, the Leishmania and the trypanosomes. The KanetoPlast reference is to an organelle that sits inside the cytoplasm of both of these groups, which performs a very unusual function. It takes the place of what ordinarily would be a mitochondria. So what we really have here is an organelle which has evolved over time to accommodate these special niches that the Leishmania and the trypanosomes have come to be selected for in the human host. The function of the KanetoPlast, as I've mentioned, is one of respiration. But it consists of two different kinds of DNA molecules. They're both circles. And the first kind are the ones that encode the proteins which are important for aerobic or facultative aerobic respiration. Those are known as maxi circles. And then there's a set of smaller concatenated mini circles, which together forms a complex which you can actually see if you do an electron microscope picture of these organisms in thin section. You can actually see these DNA molecules all integrated together in that organelle referred to as the KanetoPlast. So what is the function of the mini circles? If the maxi circles encode proteins that aid this organism in its energy metabolism, the mini circles took a longer time to reveal themselves in terms of their biological function. And what they turn out to be are guide RNAs. They produce guide RNAs, which then alter the DNA code itself, and they change cytosine to uridine. They actually edit RNA. And so RNA editing was first discovered in a trypanosome, but later on discovered in lots of other places too. So here's the origin of that knowledge that we can now say sort of sets in stone the way in which that DNA molecule is read and then produces the RNA molecules, which then go on to, of course, produce the proteins. So it's a complicated biology. We haven't got too much more time to discuss it, but just know that there are some drugs that actually target the KanetoPlast, and it'll come out later on when we discuss treatment as to how this works. So let's discuss this life cycle. And the life cycle consists of two parts. One is take place in the sandfly. So yes, there's a vector here now. So now we have not only a life cycle which involves a complicated biology with complicated organelles and complicated stages of the infection, but it also exists in two different places, in two different host species. The sandfly, which is a diminutive little fly that lives kind of unbeknownst to most people. They're very weak flyers, and there are two kinds. There's the old world variety, as we'll see, and then there's the one found in Central and South America. They both serve to transmit the Leishmania parasites in those two places. And as you can see the schematic of their gut tract here, I'll explain what that means as we discuss what happens inside the sandfly as it takes a blood meal to feed itself so it can reproduce. So in the old world, as it's called, or in, let's say, Africa, Asia, and Europe, the genus for sandflies are phlebotomous. Phlebotomous is a reference, a Latin term for drawing of blood. So a phlebotomist that has their occupation in a hospital, those are the people that you hold your arm out to that put the tourniquet on it, and they would draw some blood. If you ask those people what they do for a living, they would say, well, I'm a phlebotomist. If you ask this sandfly what it does for a living, they would give you the same answer if they could speak. So the sandflies are the way in which this infection is transmitted from person to person. But as you might be able to guess, since all of these were listed as zoonotic infections as well, sandflies are not discriminatory in terms of who they bite. They can bite a dog, they can bite a gerbil, they can bite a jaguar, or they can bite a person. And in each instance, they have the capacity for transmitting this infection. So those animals can also acquire them. Most mammals in South America, for instance, can harbor Leishmaniasis. And then in the New World, in that part of the world that I just referred to, we've got another genus of sandflies because obviously during the Jurassic period, as these two supercontinents separated from each other, the wildlife separated from each other also, and their invertebrate pathogen vectors also evolved separately. So now we have Leishmania genus as opposed to phlebotomus. There are many, many more species of phlebotomus because they've been around longer in the Middle East and in India and Africa than we have Leishmania species. We only got about 50. But as we'll see, they can cause equal amounts of damage when they start to transmit this infection. So let's talk about this parasite and what it does inside the sandfly, because it's not a simple biology. This parasite is ingested by the sandfly as it takes a blood meal. The organisms are obligate intracellular parasites, and so the sandfly takes them up in infected cells. And in this case, the cells are the very cells that we depend upon for our second line of defense, namely the phagocytic cells in our body. They're actually co-opted by this parasite and used as a site of replication. In the stomach of the sandfly, these cells are digested, and they release the amastigotes. The mastigote word refers to a flagellum, and an amastigote is, of course, an organism without a flagellum. But as we'll see, this is a stage-dependent term. So the first thing that happens is the amastigotes undergo transformation, and they grow a flagellum. They actually produce one. And now they're referred to, instead of amastigotes, they're referred to as the procyclic promastigote stage. And this stage can then divide. And it does that by binary fission, just like Giardia, eventually producing many more of its kind. Then they undergo a stage of differentiation in which they change their shape. And then they undergo a second stage of replication, finally ending up as what we would call a leptomonad stage, which refers back to a primitive stage of flagellates found in plants and other biological habitats, and which then transforms without division into the infectious stage for the next host. The next host is the mammalian host, and that stage is called the metacyclic promastigote. Lots of terms to learn, but I'll try to simplify this as we can see some examples. For instance, here, we can raise these organisms in culture. We can also raise in an insectary, the sandflies, so we can actually infect them and at various stages of the development of the parasite, isolate them from the gut tract, spread them out on a slide so you can see them. And you can see that they're all different in shape. So the procyclic promastigote stage is a little stumpy stage with a tiny little flagellum. And as that flagellum grows, the procyclic promastigote transforms into what is referred to as a short nectomonad promastigote stage. And that's shown in the next panel. That then elongates without replication into the long nectomonad promastigotes, which you can obviously see there. And they're all to the same scale, by the way, so these are comparable in terms of their sizes. And finally, the stage that's infectious for the next host species, which in this case is the mammal, the metacyclic promastigotes are shown here too. So let's just see how this diagram then plays out. So the amastigotes are shown in A, so that's taken up inside this blood meal that you can see here. And here are the amastigotes. The blood meal occupies this entire bolus of blood. And instantly, as the cells are digested away from these amastigotes, they transform into the procyclic trypanosomes, or Leishmania, I'm sorry. So let's say that again. The amastigote stage grows a flagellum and develops into the procyclic promastigote stage, which then replicates. As you can see here, the arrows indicate a replication of from one to more than one organism. They then differentiate. They then differentiate further into the short form of the promastigote stage, and you can see that shown here, and for another cycle of replication, which then results in the production of leptomonads, and the leptomonads without division then differentiate into the metacyclic promastigotes. Pretty complicated stuff. Nothing that you'd have to remember if you're a practicing physician, for instance, but it's good to know what goes on in the sandflies anyway. And so that's a brief explanation. I'm sure that there's a lot more information out there that we could talk about, but I think it's in our best interests to move to the mammal. As the metacyclic promastigote stage is introduced into the mammalian host, there are a number of elements that come into play, both from the sandfly itself and from the parasite, which ensures the successful feeding of the sandfly to absorb as much blood as it can, and also for the host not to react to the presence of foreign material like salivary secretions, which contain proteins of various sorts from the sandfly, or the offending organisms which are being injected into the site of the bite wound. We have modulating molecules that are produced by the parasites. One of them is called maxidillin. It's only a 7 kilodalton Leishmania specific peptide. And for the sandfly, at least, it's a very interesting molecule because what it does is it causes the arteriole to dilate. It makes it easier for the sandfly to insert its proboscis and draw out blood. But in fact, what it ends up doing is inhibiting the response of the sandfly to stop feeding, and it actually increases the amount of blood that the sandfly can obtain till almost the point of bursting. And this ensures, of course, that the sandfly might then acquire another infection from that same host. The Leishmania organisms also contain virulence factors which act at the bite site to prevent the release of inflammatory responses, which may inhibit the successful transfer of the promastigote stage to the cell which is about to ingest it. So things like tumor necrosis factor, alpha, and upregulating things that stimulate inflammation at the same time, because remember, these organisms infect inflammatory cells of various kinds, particularly macrophages and dendritic cells. So this organism actually stimulates the production of inflammation without the production of tumor necrosis alpha. And as a result of that, it's basically recruiting host cells for the further carrying on of its life cycle. So collectively, the organisms downregulate Th1 type immune responses, which are, from the host perspective, at least the mammalian host, protective, in favor of Th2 type responses, which, in the perspective of the parasite, are non-protective. So let's look at the life cycle of the mammalian host now. It's a little more simple than the one that's carried out in the sandfly. And we begin with the metacyclic promastigote being injected into the tissue anywhere on the skin. It doesn't matter where this occurs, but in most places where the skin is exposed, of course, that's where the sandfly will gain the best access to the host. Sandfly is taking a blood meal. All these things are going on at the same time, and there are many more biologically active molecules that the sandfly secretes into the bite wound to enable it to have a successful blood meal. And this happens very quickly. This is a matter of seconds that we're talking about. The promastigotes are released into the bite wound. They are then taken up in the subcutaneous tissues by dendritic cells, by macrophages, and the parasite actually infects these cells without being digested by them. You'll also notice depicted on this life cycle, we have reservoir hosts. We have desert-dwelling animals, such as the gerbil that you've seen depicted here, or peridomestic animals like dogs, for instance, which can harbor the same infection. So this infection, in this case, we're talking about Leishmania tropica, which is a dermal restricted organism that causes cutaneous leishmaniasis in most cases, stays within the skin itself, recruiting more and more inflammatory cells into the area, which it uses as host cells. It then reproduces at such a rapid rate that it causes the lysis of those cells. And of course, the organism then has to find another cell, which is not a problem because of the intense inflammation that occurs at the same time. Eventually, over a matter of weeks, a lesion is created. This lesion is a crater-formed lesion. The living edge of that is raised. And if you take a biopsy, you'll find that all of the organisms are at the edge of this lesion. The inner portion is filled with necrotic cells and scar tissue. For the infection to continue, of course, we need to transmit this to another sandfly. So at that point, sandflies would be attracted to a wound because the blood supply is fairly close to the surface at that point, as the repair mechanism is slow to develop, but nonetheless is ongoing. The sandfly then takes a blood meal. This is a non-infected sandfly in this case. The ingested macrophages with the amastigote stage of Leishmania are taken up, and that very complicated life cycle that I described to you earlier in the sandfly then occurs over the matter of about a week to 10 days. Here's a nice picture of what a promastigote looks like, a metacyclic promastigote with its nice flagellum. These three organisms were actually grown in vitro, and as we'll see, represents one way of diagnosing the infection. Here's a nice scanning electron micrograph showing a macrophage actually being invaded by two metacyclic promastigotes. The macrophage, of course, thinks it's going to protect the host from infection, where in reality this parasite has developed some very interesting intracellular strategies for avoiding digestion. And of course, in this case, the hunted becomes the hunter. And we see what happens. In this smear that's made from, in this case, a bone marrow smear, but it's the same process, basically. And you can see in this smear all these small little punctate blue dots. And it's a large blue dot here. That's the nucleus of the organism. And the KanetoPlast is shown here, which is a smaller blue dot. So you have a large and a small blue dot. I know that that would be very hard for an histopathologist that's in a laboratory which is not used to seeing this disease a lot to actually make the identification based just on this type of approach to making a smear from, let's say, a biopsy. And that's why we have other ways of diagnosing this infection as well. Now here's an interesting electron micrograph showing what looks ordinarily like the organism living inside of a parasitophorous vacuole, which you can see surrounding the organisms here. We have several amastigotes shown in this depiction. We also see a thorotrast labeled lysosome. So we can identify the lysosomes simply by feeding thorotrast first to macrophages and then infecting them with Leishmania. And we can easily show that Leishmania do not inhibit the fusion of the lysosome to the parasitophorous vacuole. So then we have to search for other explanations as to why this organism is able to survive in such a hostile environment. And the answer comes out to be very interesting indeed. This organism has evolved over time to counteract all of the lysosomal enzymes with an enzyme of its own. It's a cysteine protease called cysteine protease B. And this cysteine protease is able to chop up most of the proteases from the host that are released into the parasitophorous vacuole. It doesn't prevent acidification. It doesn't prevent fusion. It doesn't prevent the enzymes from actually attacking the parasite until it gets to the surface. Once it's at the surface, the cysteine protease takes over and chops them into little pieces and renders them inactive. Very nice strategy for survival inside of a cell if you're an intracellular parasite. There are other ways this parasite avoids detection or avoids destruction. One of them is to downregulate interleukin-12, because as we know, interleukin-12 is associated with the upregulation of interferon gamma, which is protection inducing. So it downregulates this. As a result, it takes a longer time for protection to develop. Amastigotes, as I mentioned, possess this cysteine protease B, which is very important for its survival. The amastigotes also release other inhibitors of lysosomal cysteine proteases from the host, which counteract their activities and essentially blocks all of the lysosomal enzymatic activity. There's another protein produced by the parasite called Leishmanolysin. It's also known as the 63-KDA metalloprotease, which enables the amastigotes to migrate to a new cell. Once they rupture out of the cell that they infect and, so to speak, use up all the resources, they are able to then rupture out of that cell and find other cells in which to infect. And that's how they infect the contiguous inflammatory response that the host generates. It does so by digesting its way through the tissues, mostly by digesting collagen type 4. The other thing it does, at the same time, this is the same protein, does two different things. And it also binds to natural killer cells. And in doing so, it inhibits the production of interferon gamma in those cells, which you might expect to come to our rescue if the second line of defense is dead. At least some part of the innate immune response, the part that's not inducible, but it's always there, at least they can come and help us. And in last-ditch stand, it doesn't seem to work out too well, at least in the beginning and middle part of the infection. But it's not hopeless, as we'll see. And then there's another group of Leishmania, only present in the New World, particularly in Brazil and in other South American countries, which are infected, or which harbor, I should say, a double-stranded RNA virus that is associated with the induction of intense hyperinflammatory response. Now, remember, this organism loves inflammation without the production, of course, of interferon gamma. So this virus actually helps it do that, and it attracts more and more cells to the area. Therefore, you get more and more infection. And it also ensures the spread of this infection because it makes more host cells available that are infected to the sandflies that come along and bite these people. But as I mentioned, it's not hopeless. A host eventually does mount a protection-inducing immune response. And they're mostly Th type 1 responses, and they're strong. They induce lots of CD4 plus T cells in terms of memory. Langerhans cells are involved in producing antigen presentation, and in presenting antigens, I should say, and in the induction of interleukin 12 and interleukin 27. Interleukin 27 upregulates IL-10, and IL-10 is associated with the production of interferon gamma. So we see how this thing can eventually wrap up the infection and prevent it from overwhelming the host. But in the meantime, it's created these large disfiguring ulcers, perhaps. Parasites are killed by activated macrophages that produce nitric oxide, as you might expect. And chemokines are also thought to play an important role, particularly IMP3 beta and interferon gamma, as I mentioned before. There are some small differences between various groups of Leishmania. The visceral Leishmaniasis group, for instance, is mostly protected by an upregulation of interleukin 12 and interferon gamma, but I must caution this optimistic approach to an organism which has an 80% mortality rate if not treated. So these immune responses, while they are present and while they do hold the parasite in check for some time, do not prevent the parasite from eventually overwhelming the host, particularly in small children and in older adults.
Now, I want to just reinforce the clinical presentation, and we're going to go more into depth, or I'm going to go more into depth when we introduce clinical vignettes for the different subtypes. But with Leishmania species, it's actually the species that determines the clinical presentation. And I like to have people think in terms of threes. There's cutaneous leishmaniasis, which is going to be something we see with a large number of Leishmania species. We're going to see mucocutaneous leishmaniasis. And that is primarily, again, going to be the rule of three, primarily three species in the Viana subspecies. It's going to be the Leishmania brasiliensis, the Leishmania panamensis, and the Leishmania guyanensis. And I will say very rarely, to break my rule of three, amazonensis has been described as well. Visceral leishmaniasis is going to be predominantly Leishmania donovani, Leishmania infantum, and there will be a few others that rarely can visualize. So as we go forward, these particular species will be brought up, and we'll be discussing how that impacts the potential for future clinical presentations and the impact that has on our clinical decision with regard to treatment. Now, the three major presentations, just to really put this in people's mind, you can see here on the far left, I'm going to bring my cursor over, this pizza pie type lesion with an indurated edge. Actually, it's nice because you can see that, and it really, induration is something you feel, a hardness. And unfortunately, this is on this young woman's face. We have mucocutaneous leishmaniasis, and this is really a horrible disease where we're having a manifestation right at the junction of the mucous membranes and the skin. This can happen on the lips. It could happen right around the eyes. It can happen in the nose, or a couple of those other sites where the mucous membranes and skin come together. And again, this is only going to be caused by certain species of Leishmania. We also have visceral leishmaniasis, predominantly an old world disease with rare exception. And in general, I'll leave you the rule of threes, but also in general, mucocutaneous disease is a new world problem, and visceral disease is in general an old world problem.
Well, we could talk a lot more about this, and we should, but we have other Leishmania to discuss. In fact, we have some specific diseases to go into now. So if you want to learn more about this, here are some interesting references that are relatively up to date. They contain various overviews of immunity, vaccine development, and perhaps why aren't there vaccines to this already? And then, of course, we've got 2 episodes on TWIP, episode 14 and 95, which you can easily access on microbe.tv/TWIP for your listening pleasure. So next time, with regards to leishmaniasis, we're going to discuss the cutaneous form and all of its manifestations.